in the process of transcription, this is called as a template. So, correct, this is called as a template strand. And the one that does not participate in the process of transcription, it is called as coding strand. Okay, so how do we recognize which is template, which is coding? It's very simple that the synthesis of mRNA always happens in what direction? It happens in 5' to 3' direction. So if there is 3' to 5' direction is the mother strand, then your mRNA will be synthesized from 5' to 3'. Okay, so this is your mRNA. Correct, right? So that is why we say that 3' to 5' strand is always a template strand and 5' to 3' strand is a coding strand. It's a very simple thing that we can use to recognize which is template and which is coding. Now let's understand the mechanism of the whole DNA, the transcription process. Okay, just give me a second. So in the transcription, what we have to do is we have to manufacture a new mRNA or an RNA sequence. So for the synthesis of an RNA, there are some prerequisites. Like you saw in the DNA replication, we involve so many proteins and enzymes to accomplish this process. Similarly, to accomplish a transcription, it requires minimum ingredients. What is that? DNA helicase. Okay, one second. DNA helicase, RNA polymerase, ribonucleotides, etc. Correct? It's very simple. DNA helicase, we know that the reason why it is required, it is unwind, right? Whereas RNA polymerase, what do we have here? So this RNA polymerase is a little bit complex. Okay, it's a little bit complicated. So that is why we keep it aside. So this role of RNA polymerase is to synthesize RNA. So there are different types of RNA polymerase, RNA pol1, pol2, and pol3. Hopefully I can find it in the slide. Just give me a second. Yeah, here it is. So let's talk about your RNA polymerase. So first one, we understand that the prime ingredient that we want is helicase to unwind the DNA strands. So here is your DNA strand, correct, guys? This is your DNA strand, and this is completely wound with each other. So to start the transcription, you have to unwind the strands temporarily, not permanently. Unwinding will happen temporarily, and that unwinding is brought about by who? The helicase. So this is your helicase. Helicase will open the strand and it will expose the single strand. Now what will happen once the strands are open? Now here the synthesis of new RNA will take place. So this is the DNA strand, this is another DNA strand, and this is the new RNA strand that is being synthesized. And second ingredient, what we understood is the RNA polymerase. So are all RNA polymerase same? Definitely no. If you see in the prokaryotic as well as eukaryotic cells, we come across similar RNA polymerase. So those RNA polymerase are classified as RNA pol1, pol2, and pol3. Okay, so you can simply call it as RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. Now, this is a very, very, very important slide in the whole process of transcription. Okay, just try to focus. And if you cannot focus, please write it down. Okay, so in RNA polymerase, we have RNA pol1, RNA polymerase 2, RNA polymerase 3, guys. So it's always asked, what is the function of an RNA polymerase? What is the function of RNA polymerase 1? What is the function of RNA polymerase 3? Usually they don't go for RNA polymerase 2. It is very simple. You understand my point? So that is why this slide is very, very important. So what type of RNA polymerase we have in the cell? We have three types of RNA polymerase: RNA pol1, RNA pol2, RNA pol3. Let's talk about RNA pol2 first, because it is very, very simple, easy, and more important for our understanding of transcription. Okay, RNA polymerase 2, where is it found? It is found in the nucleus region. And what does it produce? It produces something called as mRNA. So what is the role of mRNA? mRNA is a sequence of RNA that is responsible for the production of protein. Okay, remember in your biology, so for you to make you understand, if you look at the whole organism as human beings, right? As a human being, as an organism, if you look at, we are biological beings and majority of our functions, our expression, whatever we do is 98% based on proteins, guys. That means all the functions, your enzymatic reactions, your catalytic reactions, your biochemical reactions, your proteinaceous reactions, most of the reactions that makes you running and be like a organism is all taken up by proteins. Proteins perform 98% of all biological functions. That is why it is necessary to synthesize proteins to make the genes express. Got it? No? So the DNA holds the message in the form of genes, correct? No, sorry. The DNA holds the message in the form of genes. So that gene is converted into an mRNA. What does M stands for? M stands for a messenger. Okay, it's a message. Who does it carry a message to? I will talk about it. So this message is responsible for the construction of the proteins. You get my point? So this is in prokaryotes. A direct messenger RNA is produced in prokaryotes using the RNA polymerase 2. And is there anything that it can do? It can also synthesize in eukaryotes something called as hnRNA. What is hnRNA? Heteronuclear RNA. So to understand what is the meaning of heteronuclear RNA, we have to just wait till we come to translation. So what is the function of RNA polymerase 2? RNA polymerase 2, it is found in what region is very, very important. It is found in the region of nucleus. What is the function? It produces all pre-mRNA. Pre-mRNA is called as hnRNA or mRNA in prokaryotes, which is responsible for the production of proteins. Clear, Yes? Is it clear? Chalo. Now, what's the next one? Next one is our RNA pol 1, RNA pol 3. Very, very, very important, guys. Please give some focus. RNA polymerase 1 is found in the nucleolus. Okay. So what is the role of this RNA polymerase 1? You remember that RNA polymerase is responsible for the synthesis of all rRNAs. Okay. rRNA. What is the meaning of rRNA? rRNA means ribosomal RNA. Why are we talking about ribosomes now? Because that you studied in the first year that in the cell unit of life, the role of ribosomes, right? Ribosomes is what? Protein synthesis. Correct. Now, what do ribosomes do? Protein synthesis. Okay. Now, what is rRNA doing? rRNA is responsible for recruiting. It has to bring these machines. No, it has to bring the labors. Who is our labor? Our labor is our ribosome. So to recognize the perfect ribosome, to come and assemble for the production of protein, what do we need? We need rRNAs. rRNA, what it will do? It will go, one second, easy trick to remember the sequence. RAMAT. Now can you expand this so that even I will be able to understand? JNVR, RAMAT. I don't know what it is, but I never came across this. Maybe if your teacher has taught this, excellent, because you're able to remember this. Okay. RAMAT. Okay. Ribosomal RNA. Okay. Messenger RNA. E is what? No, you please expand for me. It's very curious. Okay. So now what it is doing, RNA polymerase 1, it is found in the nucleus. It produces all rRNAs. Okay. What is rRNAs? It is a ribosomal RNA. What is the function of ribosomal RNA? Guys, I told you, ribosomes are subunits. You see? So what do they do? So these ribosomes, which are like, we have 80S ribosome, 70S ribosome, correct? No, 55S ribosome. We have come across in the cellular unit of life. We know that there are different types of ribosomes. But how do you make the ribosomes come to work for you? So by producing a ribosomal RNA. So the role of ribosomal RNA is to recruit your ribosomes. Got it? No? But what is the one important thing that we have to understand here is our RNA polymerase 1 will synthesize all types of rRNAs except 5S rRNA. Very, very, very important. What is 5S rRNA? 5 small nuclear RNA, we call it as. Okay. So this particular RNA is not synthesized by your RNA polymerase 1. Got it? No? So you make a point of it. I mean, like you try to remember this. Got it now? So next we go for RNA polymerase 3. What is our RNA polymerase 3? RNA polymerase 3 is also found in the nucleus region. Remember, RNA pol 2, RNA pol 3, both of them are found in the nucleus region. Now, what is the function of your RNA polymerase 3? The first most important function of your RNA polymerase 3 is your tRNA. What is tRNA? It is a transverse RNA. We have to discuss the structure of tRNA, the function of tRNA, the activation of tRNA, and the details of tRNA in detail as we go further. But right now, you just try to memorize this. RNA polymerase 3 is responsible for tRNA. And apart from the tRNA, it is also responsible for 5S rRNA and also small nuclear RNAs. I told you that I will be discussing about the tRNAs in detail lately. But what about this 5S rRNA and small nuclear RNAs? So they are involved in a very specific function called as splicing. Okay, splicing. So what is splicing? What is the definition of it? What is the meaning of it? We will understand as we move further. Did you understand? Okay, you come across RNA polymerases. What is the function of RNA polymerases and where can they be found? So this is a very important slide. I hope you remember. Okay, now. So chalo, let's go for your next one, ribonucleotides. So we understood what is DNA helicase role and what are the types of RNA polymerase we have. And then we have ribonucleotides. What are ribonucleotides? I told you, for any polymerization reaction, for any biochemical reaction you take, what is required? We required energy. Especially for what? For something called as construction. What is construction? Construction is also called as anabolic reactions. For every anabolic reactions, you require energy. So where do we get energy? Energy is coming from who? It is coming from your ribonucleotides. What is your ribonucleotides? How is it different from deoxyribonucleotides and ribonucleotides? Very simple. Our ribonucleotides do not have thymine. We have ATP, GTP, CTP, and then you have UTP, uracil triphosphate. We don't have thymine triphosphate, but we have uracil triphosphate. That is the sole difference between your RNA and DNA. Are you understanding my point? Yeah? Chalo. Now, these energy molecules, in the last class, I explained how these energy molecules will help in the building of your new DNA. Similarly, the same process, ATP is converted to AMP, GTP is converted to GMP, UTP is converted to UMP. So, conversion of triphosphate to monophosphate, you require breaking bonds and you get energy. Through this energy, you can start synthesizing a new RNA. Correct? No? Let's go prokaryotic transcription and your eukaryotic transcription. Last time I told you this is a very, very important things that you should not miss. So let's start with it, and I hopefully feel that you all will understand and enjoy this. Okay, whatever is coming further. The whole transcription process, guys, can be divided into three phases. Correct? No, initiation, elongation, and termination, which you have already studied in your 12th class. I mean, like textbooks. But what is initiation? Why initiation is such an important factor or a parameter? If you look at it, first we will start about prokaryotic transcription, and then we understand the eukaryotic transcription, and we understand what is the core complexities between these two. If you see both prokaryotic transcription and eukaryotic transcription, it always goes in three phases. One is initiation, elongation, and termination. Got it? No? To start with initiation, first we are talking about prokaryotic transcription. So we start about initiation. Okay. So this class we start about initiation. So initiation. How does the whole process of transcription even begin? Can the transcription start anywhere in the gene? So our genes are very well regulated and they are very well monitored. So that is how we are able to express how much ever we want. We don't see excessive expressions in a normal organism. You only see how much is required, only that much. You see that only how much is required. You have 5 fingers, only 5 fingers. You can see the development of your characters. It's all very, very well balanced. So how is it happening? For all that, one region is very, very important. That is called as promoter. What is a promoter? Promoter is nothing but a conserved sequences. So what is conserved sequences? Conserved sequences are nothing but it is coming from one generation to another generation, another generation to another generation over thousands and thousands of years. without even getting a small modification. There is no small modifications. That is first thing. Okay, so they are nothing but conserved sequences. I can tell you the conserved sequences. So where can I find these conserved sequences? So now you have something called as upstream and you have something called as downstream. You understand my point? So what is upstream promoters? See, if this is your gene, let me tell you. If this is the gene, this is the DNA that carries all the messages. And this is the sequence of DNA that is before the genes. See, this is before, no? Correct, no? This is what this is called as after. I mean, like this is genes, let's say. This is what after. You can divide this into three different sequences, no? Correct, right? So now, what do we have here? We have before the genes is called as upstream. Against, no? STREAM, yeah, upstream. Once this is starting, like from the beginning of the gene, this is called as downstream. So whatever the numbers, for example, if this is the first pair, A, C, C. So this is what +1. This is what +2. Like this, +3, +4, +5, +6. It goes on as the number of base pairs increases. So the downstream is always represented by plus. The upstream is always represented by minus. You get my point? So how does a transcription takes place? Transcription starts here, no? Well, before the genes, it'll come and assemble. And once it comes to the genes, it will go down in what direction? 5' to 3' direction. Correct, right? So the genes, the transcriptional machinery, the machine, which is responsible for your production of your mRNA, always has to move towards what direction? Downstream, like the boat going through the flow of your river. Okay, if you go with the flow of the river, that is called as downstream. If you go against the flow of river, that is called as what? It is called as upstream. Very, very simple. Correct. No, but guys, you got it right. So now, what are promoters? Promoters are conserved sequences or ancestral sequences. Where are they present? They are present upstream. Okay, they are present upstream. If you don't understand, you can see here. So this is the beginning of the gene. From here, the +1, first nucleotide where the real genes is present, that is called as downstream. Correct? No. Here you can see. What is +1 before 0? What is before 0? -1, -2, -3, -4. Now you can see in prokaryotes at -10 and at -35 region, you can see that in every prokaryotes, there are very, very similar, absolutely same sequences. And these sequences are called as conserved sequences. And these conserved sequences are present in what region? -10 region and -35 region. What is present in -10 region? You can see that AT rich region or TA rich region can be seen. What is there? TATAAT. So that particular sequence is conserved. It is present there. And next, what do we have here? We have something as in 35 region TTGACG. So this is the same sequence you can come across in all prokaryotes. And these sequences, which are called which are called as -10 region and -35 region, are called as promoter sequence. You understand? Now, why are we even talking about promoters? What are promoters? At the upstream of your genes, there are some conserved sequences, especially in prokaryotes and even in eukaryotes. In prokaryotes, if you are talking about prokaryotic promoters, at the -10 region and -35 region, we have conserved sequences. You understand? And these sequences are responsible for what? For assembling, okay? For assembly of transcription. Correct. No, guys. How will your RNA polymerase even understand, or your RNA polymerase even know where to start the transcription process? Correct, right? There has to be a point where everything begins. So that beginning point for the transcription is your promoter. And based on what type of promoters you have, okay, your power, what do you tell? The power of your transcription also increases. You can see that Some genes are always functioning. Some genes are sometimes functioning. Some genes will not function at all. Only once or twice they have to function in their whole lifetime. So who decides that? The decision is made by the promoters they have. You understand my point? So what is the role of promoters? They become the recruiting point for your transcriptional machinery. Now, what does our transcriptional machinery include? Our transcriptional machinery includes, okay, give me a second, guys. Yeah. Our transcriptional machinery includes what? Our RNA polymerase. Okay. So what is our RNA polymerase? RNA polymerase is nothing but an enzyme. So now this RNA polymerase, it is a holoenzyme. What is the meaning of holoenzyme, guys? If you have any enzyme is a protein, correct? No, enzymes are nothing but proteins. If you have more than one subunit, if you have more than one subunit, it is called as a holoenzyme. Now let us talk about your RNA polymerase 2. Okay. What is your RNA polymerase 2 do? RNA pol 2 will produce your mRNA, correct? No, guys? It will produce what? mRNA. Now let us see what does our RNA polymerase 2 is made up of. RNA polymerase 2 in prokaryotes, it is made up of 5 subunits. What are the 5 subunits? Alpha alpha, beta beta dash, and your sigma factor. You might have come across your sigma factor, correct? No? What do you call the sigma factor? You say that in your textbook, it says that the transcription begins at a place called as a promoter sequence. The promoter sequence is recognized by your sigma factor. The sigma factor comes and attaches to the promoter sequence, and then it recruits the rest of the RNA polymerase. So once this RNA polymerase is assembled, now the initiation of transcription is completed. Now, with the use of ATP, the transcriptional machinery jumps from the promoter sequence all the way till the structural gene. Then the elongation process will start place. This is what you have learned in your textbook, correct, right? Now, we understand what is this RNA polymerase. I told you RNA polymerase is nothing but a holoenzyme. What is it? It is a holoenzyme. Very, very important point. Why we call it as holoenzyme? Because it is not made up of one particular polypeptide. It is made up of 5 different types of subunits. What are the names of subunits? Alpha alpha, beta beta dash, and sigma factor. Got it? No? Now, what is happening in this whole transcription? We know that first, initially, our sigma factor will come. Okay. So what are our subunits? Alpha alpha, beta beta dash, and then you have your sigma. Sigma is the one that recognizes the promoter. It comes and it sits in the promoter sequence. And then what it will do, the reaction that is recognized by the sigma will start recruiting other, other subunits. So what are your subunits? You have your alpha subunits and beta subunits, correct, right? Let's talk about alpha subunits. What is the role of alpha subunit? It helps in assembly of polymerase, all the rest of the polymerase on the DNA. What is the other polymerase, or what is the other subunits are doing, like beta subunit? Beta subunit is responsible. Now you can see here, wait, I think I have it here. You can take a note of it also. Where is it? So, it's not, okay. Let me write it here. I know this. Okay. Beta subunit, what it will do? Beta subunit is responsible for RNA and DNA. It will clamp. It will hold it together. Okay. Now, what is beta dash doing? Beta dash is responsible for converting of all RNTPs. Okay. And it will convert it into RMP, ribonucleotide monophosphate. That means it is responsible for energizing the whole reaction. Now, the details of the subunits are not necessary. Now, what you have to understand is the sigma factor will come and assemble. And then, see, this is your genes and this is your promoter sequence. Okay, this is your promoter sequence. Sigma factor is the one that recognizes the promoter sequence. It will come and sit here. Once the sigma factor comes and assembles, what will happen? This will create a chain of reactions. That means, okay, sigma is here. Let me come alpha. And another alpha is recruited. And then another beta is recruited. Another beta dash is recruited. Now, what will happen? All the subunits of your RNA pol has assembled at what region? At the promoter sequence. So once this assembly is done, now the sigma factor job is finished. So when its job is finished, now what will happen? The sigma factor will come out. Once the sigma factor will come out, your alpha alpha, beta beta dash will complete the assembly of what? RNA polymerase. So once this assembly, when does the assembly get completed? Only when the sigma factor comes out. When the sigma factor comes out, these four will produce a functional enzyme. What is our functional enzyme? RNA polymerase. So once this functional enzyme has been assembled, now what will happen? Now look here, guys. Look here. This is your genes. This is your promoter. This is your structural gene. This is your terminator. Correct? No, this is your structure of a gene. This is your terminator sequence. Here is where your RNA polymerase has assembled. This is where your first base pair is located. So how does it move from here? Now a huge burst of energy will take place. It will utilize ATP. Okay, it will utilize ATP and then it will convert into AMP. With this energy, what it will do? The whole enzyme assembly will jump from promoter sequence all the way till the structural gene. Just a jump, a huge jump will take place of the RNA polymerase. Once they have come here, now what happens, guys? They can start synthesizing a continuous sequence of your RNA from 5 prime to 3 prime direction. Did you understand? So what did we understand in the initiation? In the initiation, we understood that we require something called as promoter sequence, and the promoter sequences are conserved sequences that are present in the gene. So where are they located? If this is the beginning of the gene, before the gene is called as upstream. So the upstream regions are called as a promoter sequence. In prokaryotes, we have -10 region and -35 region. So this is about promoters. What about RNA polymerase? What is RNA polymerase? RNA polymerase is nothing but a holoenzyme. Why do we call it as a holoenzyme? If I take a prokaryotic as an example, in prokaryotes, RNA polymerase is made up of several subunits of polypeptides. What are the subunits? Alpha alpha, beta beta dash, and then you have sigma. Each of them have very, very specific roles. So how is this subunits assembly take place? The promoter sequence is recognized by your alpha sigma factor. The sigma factor will come and assemble at the promoter sequence. Once the recognition of the sigma factor take place, the other subunits will start assembling onto the promoter. So once all the subunits are in place, then what will happen? The sigma factor will disassociate. That means sigma factor will come out. Once the sigma factor comes out, your alpha alpha, beta beta dash will produce what? A functional RNA polymerase. Once they produce this functional RNA polymerase, now what they do? They jump from the promoter sequence all the way till the structural gene. How does the jump take place? With the utility of energy. Once they have jumped here, that completes something called as initiation. Did you guys understand this? Yes, no? Are you able to hear me, first of all? People. Are you able to hear me, guys? Are you able to follow what I'm trying to say? Is it easy? Yeah. So just put it in the chat box, okay? Sure. So once the initiation is complete, the elongation is very, very simple. How does the elongation take place? Elongation always takes place in the direction of 5 prime to 3 prime. And for the elongation, what will happen? You require ribonucleotide triphosphates, okay, RTPs, which is called as ATP, GTP, CTP, and then UTP. All this will come across your your RNA polymerase, and this RNA polymerase will use this all the triphosphates, and it will start constructing your mRNA in 5 prime to 3 prime direction in a continuous fashion. Done. Now, what happens with the termination? The termination is very, very exciting. So you have two types of termination. One is a row dependent termination. Other one is a row independent termination. Since the explanation is so much here, you can take my PPTs and you can study. But for now, I will start with the or explaining you guys. So what is termination? See, this is your, oh yeah, excuse me guys. This is your gene. Here is your promoter sequence. We understand what is the role of promoter sequence. Here is your structural gene. Okay, sorry, why it's going cross. Here is your structural gene. And after finishing of the structural gene, what do you get? You get termination. You have to end the process of transcription. You can't keep on going on. Forever, the length of the gene will not be there. At one point, it will start. At one point, it will end. So once it has end, there is no point of your transcriptional machinery. What is your transcriptional machinery? Your RNA polymerase to move further than this, a structural gene. So to stop it, there is a mechanism called as termination. What is termination? The end of transcription. So how is this termination sequence designed? So the termination machinery is designed into two different types. One is called as row dependent. Other one is called as row independent. You get my point? What is row dependent and row independent? We see here. So this is your termination sequence. So this is your termination sequence. And in the termination sequence, imagine that you have a lot of G rich region. What is this GGG? GGG is nothing but your nucleotide guanosine. Now, your RNA polymerase is moving in a certain pace, a speed. Okay, it is moving in a certain speed. Now, if you have G in one strand, what will be the other strand? It will be C. So here, if you have G, it will be triple bond what? C. Here again, G, triple bond C, correct, right? Now, your RNA polymerase is moving at a certain speed. Now, following your RNA polymerase is a protein called as Rho protein. This protein is called as what? Rho protein. Now, Rho protein is very bad in traffic management. How? It does not have any control on the brakes. Now, imagine your RNA polymerase is going in a certain speed. Suddenly, it comes across a very high energy rich region. What is very high energy rich region? Your gynosin region. Why it is called as high energy rich region? You can see that there are triple bonds here. So the breaking of triple bonds require high energy. Correct? To break the triple bonds, you require what? Very high energy. Now, suddenly your helicase also slows down. With the helicase slowing down, your RNA polymerase also slows down. Once the speed of the RNA polymerase slows down, this Rho protein, which was following your RNA polymerase, with the sudden slowing down of your RNA polymerase, they will start colliding. So once your Rho will collide with your RNA pol, what will happen? That will lead to disassembly. Why the disassembly will take place? You know that proteins are very sensitive to their structure, correct? No? If Rho protein collides, their structure will get destroyed. With the destruction of the structure, all the subunits like alpha alpha, beta beta dash, will all of them will dissipate. Means they will disassemble. Once they disassemble, there is no functional RNA polymerase anymore. Okay. Once this RNA polymerase is released, your mRNA, which is also synthesized, is released. Okay, the mRNA is also released. So this is one type of termination. What is this termination called as? This termination is called as row dependent termination. Now, what is row independent termination? Row independent termination do not have Rho protein. Okay, no Rho protein. Then how does the termination take place? The termination takes place through inbuilt termination sequences. What is inbuilt termination sequences, guys? Inbuilt termination sequences means within the gene itself, there are certain sequences that decides the termination. So let us see how this will take place. First of all, what do we have? Let's say that we have a gene and suddenly Suddenly we have GC rich region. GC, 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 GC. Okay. And followed by, okay, sorry, I don't have enough space here. I think I will have to minimize my, this is the gene. Suddenly what it became? GC, 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 GC. And followed by that, you have AU, AU, or AT, AT. I will write it as AT, 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 rich region. Got it, no? Now, what will happen? You have your RNA polymerase, and the RNA polymerase is coming to transcribe. So, what will be your RNA sequence here? CG, 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 CG. Okay, since there is a lot of CG, now what will happen is this particular RNA will start folding upon each other. So imagine this line being folded like this. When it folds like this, you can see here, they will start creating a bonds between the, see, this is the straight line, and the straight line, what happened? It started bending over. So once it started bending over, here you have GC, 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 correct, right? So what happens here? Imagine here C comes, here G comes, here C comes, here G comes, and here C comes. Now what will happen? They start making bonds with each other. And this resembles like a hairpin. Okay, this resembles like a hairpin loop. So this resembles like a hairpin loop structure. So now this GC rich region, they start becoming a hairpin loop structure. So once they become hairpin loop structure, now you can see that further end, there is low energy. This is low energy. This is high energy. Why this is high energy? Because there are triple bonds. Why this is low energy? Because there is only double bonds. AU, 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 AU. So this AU, AU tail. The moment the polymerase comes from GC, the GC will start making a hairpin loop, and they will start forming bonds with each other. And as immediately from high energy, they go to low energy suddenly. Now what will happen? The whole machinery, your RNA polymerase, will become highly unstable. Okay. Once they become unstable, the dismantling of the RNA polymerase will take place. So once they disassemble, okay, the inbuilt termination sequences, their role is completed. You can see this is the mRNA, and this is your hairpin loop, and this is the low energy sequence. So by this way, your termination sequence is complete. Got it? No? So this is called as your prokaryotic termination. So what did we understand? We understand the prokaryotic initiation, elongation, and termination. Do you guys have any doubts? Or did you understand? Can you please confirm? If you have trouble in telling me through your chats or something, let me launch a poll. Okay. So I have launched a true or false. Okay. If you have understood, put it true. If you did not understand, put it false. Now this will make me easy to understand. Yeah. Thank you guys. Thank you for the response. It's very sweet. Okay. Shalom. Yeah. The one person who did not understand, you're always free to ask me if you do not understand. As I told you, there is no barriers here. The only reason I'm here is to help you guys. Okay. Chalo. Now let's talk about eukaryotic transcription. If you look at eukaryotic transcription, guys, it's more complicated. It's even more complicated. Sir, do we have to even study eukaryotic transcription? For one reason, you have to study. It's not for many reasons, but for only one reason, you have to study about eukaryotic transcription. And why is that you have to study this? Let's look at it. Okay. So in eukaryotes, what happens? Even in eukaryotes, you have promoters. Okay. And then you have something called as basal transcriptional factors. Okay. Let me look at it. We don't have it. So two things are very important for us to understand here. One is something called as basal transcriptional factors, and the other thing is called as the promoters. First, let's talk about the eukaryotic promoters. Eukaryotic promoters are very similar to that of your prokaryotic promoters. In eukaryotic promoters, you have something called as starter box, cat box, octamer regions, etc. What are these starter box, cat box, octamer region? Very, very similar to that of what we saw in 10 regions and minus 35 regions. In Tata box, TATA rich region is present. In your cat box, you have CAAT region is present. In the octamer box, you have GC rich region is present. So they are all conserved sequences. So these are your eukaryotic promoters. So the eukaryotic famous promoter is what? It is your Tata box. What is your Tata box? Not our Ratan Tata. It is your TATA. That is adenosine and thymine rich region. So that is your Tata box. Got it, no guys? So this is first thing you have to remember. So the initiation of your transcription in your eukaryotes comes through the like we in prokaryotes we saw 5 subunits, right? Like alpha, alpha, beta, beta dash, and then sigma factor. Like that, in prokaryotes we don't have only five, you know, subunits. We have more than 10, more than 10 subunits. Do we have to remember all of them? 100%. I'm just kidding. Definitely no. We just have to understand the concept. Okay. So these subunits, okay, which are responsible for assembly of your transcriptional machinery are called as what? Basal transcriptional factors. BTF. Okay, basal transcriptional factors. Okay. So what are these basal transcriptional factors? These are called as TF. They are called as what? TF. Imagine we are talking about RNA pol 3, RNA polymerase 2. Let's talk about 2 because it produces what? mRNA. So since we are talking about RNA polymerase 2, what kind of transcriptional factors will they have? So I can denote them as TF2. So transcriptional factor 2. Why 2? Because I'm talking about RNA polymerase 2. What happens with transcriptional factor 2? You have, I told you, tens of transcriptional factors are there. What are those? It goes with A, it goes with B, it goes with D, it goes with E, it goes with F. What am I talking about? Transcriptional factor to A, transcriptional factor to B, transcriptional factor to D, transcriptional factor to E, like F. Like this, there are several transcriptional factors. Now, why are these factors are very important? To understand these transcriptional factors, we have to concentrate on one transcriptional factor called as 2F2D. What is 2F2D? Like your sigma factor, even TF2D is one of the transcriptional factors. Now, what does this TF2D will do? The transcriptional factor 2D has a special capacity that it has a binding site. Binding site. Binding site for what? It is called as Tata box binding protein. Very, very crucial. So now the 2F2D, what it will do is it will try to recognize your Tata box sequences, which is at the minus 30 upstream of your eukaryotic promoters. So now what will happen? TF2D is a transcriptional factor. It will come and sit on the promoter region. Once the TF2D has recognized the promoter region, now the Tata box binding protein will come and attach to this TF2D. Okay. Now, once the TF2D and the Tata box binding proteins are in place, it will start recruiting the further subunits like TF2A, TF2B, TF2D, TF2E, like this. Each of these transcriptional factors, they have a specific role to play. Now, what does it lead to? The assembly of all these transcriptional factors will lead to the formation of a functional RNA polymerase 2. So once we have a functional RNA polymerase 2, what happens? Your Tata box binding protein will disassociate. It will move away like your sigma factor. So once it moves away, this whole transcriptional factors will form as a functional mRNA, and then it will jump from your promoter sequence to your structural gene. From then onwards, your synthesis of your mRNA will take place continuously. Are you following, guys? Did you understand this? This point is very, very important. Did you understand the difference? Again, I will check it. Because you guys are not saying me anything, I have to launch polls like this. Now you know the drill. If you understood, true. If you did not understand, false. Come on, rest of you. Yeah, time is up. Okay, chalo. Got it, right? So this is the initiation in our eukaryotes. Eukaryotic transcription is very, very complex. But just to to understand a little bit, we understood how the promoter sequence is. What is a promoter sequence? They ask what is Tata box. Tata box is a promoter sequence. Now you easily understand who are Tata box, whether it is prokaryotes or eukaryotes. We know that eukaryotes have Tata box. And what are transcriptional factors? We know that RNA polymerase is not just one enzyme. It is a compilation of several subunits. It is a holoenzyme, we understood. Correct, right? So the energy for this transcription comes from your RNTPs. Correct, right? So helicase will unwind. Only one strand will participate in the process of transcription. In termination, we have two types, row-dependent and row-independent termination. In RNA polymerase, we have three types, Pol 1, Pol 2, Pol 3. The functions of RNA polymerase and where it is found and what it is role, we have understood. Okay. Now, next class is even more complex. Why? Because I'm going to talk about what is your mRNAs, what are different types of mRNAs, what is the role of each mRNAs, how are they produced, what is their initiation, termination, and elongation sequences. All of this will be discussed in the next class. Don't worry, it's going to be only 15 minutes of discussion. Then we can go for translation and genetic codes and the rest of the topics. Okay, chalo guys. So thank you very much for attending the session. As understood, very much fine. I'm very happy. If you did not understand, as I told you, you are very free to ask me any point. Got it? So chalo. Only way you can help me is by being frank with your understanding. Please attend the poll, and after that you can leave the session and you can attend the next class. Thank you very much.
Thank you, sir. Bye, sir.
Bye. Thank you.